Hello everyone, welcome to your partner in education, Agile Rank Mate. Today we're excited to start off a new series titled CUET Gateway. Now this here is a series which will be focusing on the Central University's entrance test, which is a very important examination in terms of pursuing undergraduate degrees in various central universities across India. So today, this exam is quite varied, it's quite diverse, it's got various subjects in it. Today we're going to be looking at some sample questions for this exam in the subject of biology. So, let's start off with our first question. The triplet codons UGA, UAG, and UAA are termed as termination codons because they A. do not allow ribosomes to bond with mRNA, B. do not specify any amino acid, C. prevent binding of tRNA anticodons with mRNA, D. stop mRNA synthesis. So let's solve this question. The three codons given here, UGA, UAG, and UAA, are all part of the mRNA sequence of humans. So basically, since these are part of the mRNA, they do not have anything to do with, the binding for mRNA or the stopping of mRNA synthesis. So therefore, options A, C, and D are incorrect. The right answer is option B. They do not specify any amino acid. Now, in humans, the DNA gets transcribed to messenger RNA, and then the mRNA along with the tRNA gets translated to proteins, or more importantly, amino acids. Amino acids then combine together to form proteins. Now, option B, do not specify amino acid, is the correct reason why these are termed as termination codons. So in the sequence, if the particular codon UGA or UAG or UAA comes into play, then that particular codon does not code for any amino acid. And when it does not code for any amino acid, the process of translation is stopped. Because since these do not specify anything, then there is no amino acid to attach onto this continuing structure, so therefore the process of translation stops. Then it has to be restarted by the start codon. So therefore option B is the correct option. Now let's look at another question. Segment of single-stranded RNA less than 1500 NTS that remained associated with another virus for its replication and causing of various diseases are commonly known as satellite RNA, helper retrovirus, microRNA, heterogeneous RNA. Heterogeneous RNA is what is produced in transcription, where the DNA is transcribed to form messenger RNA. And this heterogeneous RNA will help in translation to amino acids. So therefore option D is the wrong option because these genetic material do not cause diseases and they're not part of viruses. Option C, microRNA. MicroRNA are characterized by being small, being single-stranded, and they are also non-coding. So therefore they do not code for any disease or any trait. Their use in the human body is for the process known as RNA silencing. So basically, if you need to silence a trait on your mRNA strand, you use microRNA to form to stand alongside the particular code, and then that code, since it has two copies, it does not transcribe it. It does not translate itself. So therefore, the microRNA helps in RNA silencing. And this is used for fighting diseases for the tobacco plant and so on. So option C is also incorrect because this is a form of immunity and not exactly a disease. Option B, helper retrovirus. What is a helper retrovirus? This is a virus. It allows this virus allows other deficient viruses to replicate. So you have particular kinds of viruses which, is a prerequisite for another kind of virus to propagate. For example, hepatitis B is a helper retrovirus for the virus known as hepatitis D, and so on. So therefore option B is incorrect because the helper retrovirus does not attach to another virus in order to replicate itself. It is the agent that allows the other virus to get replicated. So option B is incorrect. The right answer is option A, satellite RNA. Satellite RNA is single-stranded and it's associated with other viruses in order to replicate itself, and it causes various diseases. So therefore satellite RNA is the correct option for this question. Let's look at the next question. Which of the following ecological pyramids will be inverted in shape? Now, what is the idea behind having an inverted pyramid? A pyramid, or an ecological pyramid specifically, is inverted when more number of species, or let's say more biomass or something, present on top. So for example, an inverted pyramid would look like this, and the top layer would have more species or more amount of biomass or more of the particular category than the ones below. Usually a pyramid is upright where the top tier is lesser than the tiers below it. Now let's look at our options. Option D says ecological pyramids of number of a grassland ecosystem. So if you look at the number of species on a grassland ecosystem, you have grass as the producer, then you have herbivores as the primary consumer, then you have carnivores as the secondary consumer. And if you look at a grassland, there's more grass than herbivores, and there's more herbivores than carnivores. So therefore what we have is an upright pyramid. So therefore option D is incorrect. It's not an inverted pyramid. Option C, ecological pyramids of number of a pond ecosystem. Again, same thing. You have plankton, then you have small fish, and then you have large fish. So there's more plankton than small fish, there's more small fish than large fish. So therefore this is again an upright pyramid, so therefore option C is incorrect. What about option B? Ecological pyramids of biomass in a parasitic food chain of a tree ecosystem. Now in a tree ecosystem, the parasitic food chain goes like this. It goes trees, then herbivores, and then parasites. So when it comes to biomass, trees have more biomass than herbivores. Parasites have the least biomass. But basically mass refers to the mass of the organism itself. So trees have the higher biomass, parasites have less. So therefore again this goes as an upright pyramid, so therefore option B is incorrect. The correct answer is option A, ecological pyramids of number in a parasitic food chain of a tree ecosystem. So you have one large tree feeding a small amount of herbivores, which then feeds a whole multitude of parasites. So therefore the pyramid for this particular scenario, number in a parasitic food chain, happens to be inverted. There are a few trees feeding a large amount of herbivores, which feeds a multitude of parasites. So therefore option A is the correct option. Now let's look at this question. When the enzyme ribulose 1,5-bisphosphate carboxylase oxygenase, or Rubisco for short, fails to distinguish its substrates CO2 and O2, condition is referred to as cellular oxidation, C3 photosynthesis, C4 photosynthesis, photorespiration. Now let's look at each of the options. What is cellular oxidation? In cellular oxidation, the electron donor gets oxidized and oxygen gets reduced, and this forms CO2 plus H2O plus energy. So basically, this cellular oxidation is an important aspect of photosynthesis itself. This is where water and carbon dioxide are formed. I mean, it's not a part of photosynthesis, it's a part of respiration because carbon dioxide is a product. So option A is incorrect, because the reason being that there is no use of Rubisco in this particular process. Option B is C3 photosynthesis. This is basic type of photosynthesis where CO2 and O2 concentrations vary. So sometimes CO2 is concentrated, sometimes O2 is concentrated. Option C, C4 photosynthesis. This is the advanced type. And this is where CO2 concentration is more all the time. C4 photosynthesis is structured in such a way that there's more CO2 than O2 at all times. Now, in both B and C, the enzyme Rubisco is present. However, in C4 photosynthesis, it distinguishes between CO2 and O2. So there's more CO2, so therefore Rubisco does not fail to distinguish it. In C3, there may be cases where it does not distinguish O2. However, those processes are called photorespiration and not C3 photosynthesis. So therefore, the correct answer is option D, photosynthesis. In C3 photosynthesis, you do have cases where CO2 Rubisco fails to recognize its substrates. However, most of the time, CO2 is recognized. Now, why does this phenomenon occur? The reason is that Rubisco, the enzyme, has both CO2 and O2 as its substrates. So therefore, the way in which they bond is what we call competitive bonding. So in competitive bonding, the amount of substrate that's bonded is dependent on the relative concentration. So if there's more CO2 than O2, then Rubisco bonds with CO2. However, if there's more O2 than CO2, then Rubisco bonds with O2. And when Rubisco bonds with O2, it fails to distinguish its substrates, and thus there is the process known as photorespiration, where there is a cycling of electrons. However, there is a loss of energy. So therefore, option D is the right option. Now let's look at the final question for the day. Fetal hemoglobin consists of 1 alpha and 2 beta chains, 2 alpha and 2 beta chains, 2 alpha and 2 gamma chains, 2 beta and 2 gamma chains. Now let's look at each of the options. 2 beta and 2 gamma aren't found in humans, so therefore option D is incorrect. Fetal refers to the fetus, and we're essentially dealing with the human system. So therefore, any combination that's non-human is incorrect. Option A goes for 1 alpha and 2 beta chains. Now this is incorrect because hemoglobin is a tetramer. What it means is that it needs 4 chains at all times. So in option A, you have 1 alpha and 2 beta chains. This is actually a case for alpha thalassemia, because in alpha thalassemia, 1 alpha chain is missing, so therefore option A is incorrect. Option B, 2 alpha and 2 beta chains. Now this is the most common human structure for hemoglobin. However, 2 alpha and 2 beta chains are found in adults, and not in a fetus. So therefore option B is incorrect. The right answer is option C, 2 alpha and 2 gamma chains. Now the reason why a fetus has 2 alpha and 2 gamma chains is because this structure bonds greater with O2. So basically it has a greater bonding power with oxygen. So therefore, since there's lesser oxygen in the fetus, so therefore it needs a higher it needs a compound, it needs hemoglobin with stronger attraction to O2, and that's why they use and that's why alpha and gamma chains are present in fetal hemoglobin, whereas adults have alpha and beta chains in their hemoglobin. So therefore option C is the correct option. Now that concludes this episode of CUET Gateway. We hope you found this episode interesting. For more of our useful and interesting content, don't forget to subscribe to Agile Rank Mate, your partner in education. If you want to get the latest updates from CUET Gateway or from any of our other playlists, then please don't forget to hit the bell icon that's present below the video and set it to all. That's all for today's video. Take care, stay alert, bye-bye for now.